You can see the full screen, correct?
Yes,
Yep,
looks good. Yep.
Okay. So hello, my name is Shahan Derkar-Abadian. As mentioned, I'm a postdoc at Harvard. And what I'm going to talk to you today about is essentially a little bit about discovery and discovery approaches in general for species delimitation. But then I'll get into some lecture and demonstrations about some unsupervised machine learning algorithms, which I've used to help delimit species. So I'm an organismal biologist. Of course, I have interest in anything that helps me describe species or do my taxonomy. And as mentioned, I focus on arachnids, but specifically this group of arachnids called Apiliones. So I've been thinking a lot about species delimitation the past few years because, you know, as a taxonomist, I have to describe these species. And within Apiliones, there's two big reasons why I do a lot of species delimitation. And one is that there's still a lot of undescribed diversity in Apiliones. Of these four pictures, two of them are undescribed. And I don't know if you can see the pile of jars behind me in the boxes, about a quarter of these species are undescribed. So there's a lot of work yet to be done in basic alpha taxonomy of Apiliones. And the other reason I get into species delimitation is because of the biology of these organisms. So as Brian mentioned, there's a lot of difficulty in delimiting species when they have a lot of high levels of population genetic structure, and Apiliones are no exception. So there's a lot of oversplitting, overestimating when we're using kind of the common species delimitation methods. And there's a lot of cryptic speciation in these groups, a lot of morphological conservatism. So there's a lot of difficulties. And so the reason why I got into machine learning was to try and find different ways of delimiting species that maybe don't have as much difficulty in taxa like this. So onto species delimitation. And I'm going to take a very simplistic approach in how I talk about species delimitation because I'm doing the discovery-based approach, right? So in this case, species delimitation is very simply dimensionality reduction and clustering, right? It's kind of in the name itself. Giving some group of organisms a species name is dimensionality reduction, and delimiting implies drawing lines between things. So certainly with discovery-based approaches, this is essentially what species delimitation is. And in some cases, I would argue that a lot of the validation approaches are also dimensionality reduction and clustering. So what do I mean? We have these two things, right? These things have this almost infinite number of characters you can look at, like the color of the eyes, the length of the tail, the color of this particular hair here, the color of this particular hair here, it goes on, right? But we can break these two things down into a word, a cat and a dog, right? That's our dimensionality reduction. So when we see other things that are similar to a cat and a dog, we can kind of group them together and then draw a line. That's our clustering. And of course, we can do this with genetic data as well. In my mind, sequencing a gene or 100 genes is essentially dimensionality reduction. And then of course, you can reduce those dimensions further and do some clustering and then draw your line, right? So in essence, this is what species delimitation is. So I'm going to talk about discovery-based approaches. These are approaches that serve as kind of a first step, a starting point to identifying species-level hypotheses, which are then tested in validation analyses. So discovery-based essentially just means you have no a priori hypotheses. And there are many different types of discovery-based approaches. I'm going to mention a few of them before I get into unsupervised machine learning. So one of the most commonly used methods, of course, is morphology. You know, sitting at a microscope for hours and looking at specimens, you can, you come up with certain characters that help you group species or group specimens together. So looking at this particular group, so I apologize if you hear some construction going on above me. I can't do anything about it, but if you hear it, I apologize. So looking at these specimens, you can look at some characters to help separate them. So for example, you could look at spines on the front margin of the body. This one has 3, this one has a lot more, and this one also has a lot. You can also look at color. these two are brown, this one is bright green. It's pretty amazing. This is this one you find only on moss, on tree trunks in New Zealand. It's pretty cool. So anyway, we took these multidimensional organisms and broke them down to two characters, 2 dimensions. We can then sort the specimens according to these characters and then draw our lines. And that is our clustering. This is kind of the most common way of delimiting species, even with all this genetic data still. Another form of morphological data is morphometrics. This could either be linear measurements or even geometric morphometrics. And so you collect a bunch of morphometric data, just kind of continuous variables, and you put it into principal components analysis, and then you plot it, right? And this sort of method can also be used in any other data that can be expressed as numbers, like bioclimatic variables or even behavior, anything that can be expressed as numbers. So this is kind of a very simple way of assessing morphological variation within a particular group of organisms to assess if there's any kind of significant variation. And one of my favorite morphometric papers was this one by Ezard et al. In 2010. And they kind of introduced algorithmic approaches to morphometric analyses. So instead of just doing your principal components analysis and then kind of eyeballing what you think are different clusters, they provided algorithmic and objective ways to identify species. And they do it across multiple steps. So objectively identifying the number of principal components to retain, the number of clusters, assignments to the clusters, and even outliers. So this was a pretty kind of cool way of using morphological or morphometric data to kind of help with species delimitation. So kind of before this, and one of the common things that was done was to do a principal components analysis and then draw some oddly shaped circles around these specimens and say they're different, right? I'm throwing one of my papers under the bus right now, but that's okay. So instead of kind of subjectively defining these clusters in morphometric space, you can instead use these algorithmic methods like the Ezard et al. Approach to objectively define morphometric clusters. And this was a follow-up paper to the one I just kind of dismissed, where we use morphometric data and identify these morphometric clusters objectively. And for the most part, they kind of correspond to final species based on morphology and genetic data in various analyses. So I really like this approach. I liked it so much, I ended up writing a wrapper for this. It's in R, implemented in R, and you find like nice output like this. So if this is something, if you deal with morphometric data, I would urge you to kind of use objective ways of identifying clusters and number of principal components to retain, all of that stuff. Okay, so that's morphometrics. I'm going to transition to genetic data now. You'll probably hear about some of these later in this workshop, but I'll kind of give a brief introduction to a few of the commonly used ones. So you've probably heard about ABGD. This is automatic barcode gap discovery. This is with barcode data, so CO1. And the idea behind this is that you take all your barcode data and you produce a distribution of pairwise genetic differences between all the samples. And the idea behind this is that theoretically, there should be a break between intra-species pairwise differences and interspecies pairwise differences. And you can use this theoretical break to identify that break in empirical data to identify species. Now, you know, there was a conversation about that WASP paper, and I think it's a start. I don't think it should be the only way. So I guess that's kind of my opinion. So it's a good place to start when you have, you know, if you're doing like meta barcoding and you just want to figure out what's there and preliminary hypotheses, it's great. Other methods that you might hear about. So this is sequence-based. You use the actual sequence data. Here are some tree-based methods. So PTP and GMYC. With these, you put in your tree. And in the case of PTP, it models speciation events in terms of substitution. So you would use something like a Raxamel tree. In GMYC, it's a likelihood model where you kind of fit within and between species branching patterns. And this requires an ultrametric tree. So for example, something from Beast. These, of course, have Bayesian implementations and newer versions, but these are kind of the base starting points for these methods. Okay, one of the most commonly used approaches for genetic data is structure. So I mentioned the distinction between discovery and validation. There are some cases where I think that line is blurry, and structure is one of them for me, because you can, with structure, so what it does is it's a Bayesian approach, and it assesses the pattern assesses patterns of genetic structure amongst a set of samples, assigning samples to clusters and giving proportional probabilities. So in cases of like admixture or hybridization. So you test a set of hypotheses. You can either test a limited set, which would make it kind of a validation approach, or you can test every possible hypothesis, which in my opinion is more of like a discovery. Either way, this is a good starting point to determine potential clustering patterns in your data set. Now, structure was designed for identifying populations in Hardy-Weinberg equilibrium, but it gets some use at the species level too, because ultimately you're specifying what hypotheses you're testing, what K values you're testing. So, you know, if you only test species level hypotheses, it's going to give you a species level answer, right? So structure, in my opinion, is a excellent approach. It's a dimensionality reduction approach. So it'll tell you the optimal clustering patterns and number of clusters based on likelihood. So you test your range of K values, and you use likelihood to compute the optimal K according to some statistics. One of the other commonly used methods is DAPC, and this is one I'll talk about in the demonstration. So DAPC is discriminant analysis of principal components, and discriminant analysis essentially is a way to classify samples into non-overlapping groups. DAPC was specifically kind of adapted for genetic data, and it uses structure format, which I'll show you in the demonstration as well. One of the benefits of this approach over structure is that it's much faster. Structure can take a very long time, right? You have to test all these K values, and you have to test them multiple times to get a good representation of the variation around likelihoods. DAPC is pretty quick. So, and in addition to doing principal components analysis and seeing this plot, you can also determine the number of clusters in any given data set using Bayesian information criterion score. It also kind of closely approaches structure output in terms of assigning proportional probabilities for kind of more complex data like this. Okay, so those are kind of the more commonly used discovery-based approaches. It's not all of them, of course. There are plenty of others that I didn't have time to talk about, but kind of wanted to serve as an introduction to the rest of what I'm talking about. And that is kind of machine learning. So I kind of have this boundary here that, okay, now I'm talking about machine learning, but that's not really true, because that's also blurry when you get down to it, because principal components analysis is a type of unsupervised machine learning. K-means clustering, which is used in DAPC, is another type of unsupervised machine learning. So this is the line between statistics and these methods we've used and machine learning is kind of blurry, of course. So as I mentioned earlier, the organisms I work on are difficult to delimit using genetic data only. So they're prone to morphological conservatism, deep genetic structure within species, and cryptic speciation. So many of the methods I was using were failing in these systems, right? These organisms violate the assumptions of those models. So I jumped into machine learning. I tried some different approaches that maybe find some alternatives to help me delimit all these species that are sitting on my desk. One of the benefits about machine learning is that they do dimensionality reduction. And so when looking at dimensionality reduction techniques, two-dimensional representations are the most common. So like principal components analysis, taking high-dimensional data and putting it into 2 dimensions. And machine learning methods do all of that, and they do it well. So when thinking about species delimitation, there are generally two types of analyses. I mentioned discovery-based, where there are no a priori hypotheses, and this is where you identify preliminary clusters. And these preliminary clusters serve as testable species level hypotheses, which are then sent to the validation phase and tested, where you use methods that have some biological model to inform species limits. So this is kind of the basic dichotomy in species delimitation analyses. Now, when you bring in machine learning, there are two different types of machine learning. You have, in general, unsupervised machine learning and supervised. Unsupervised uses unlabeled data and is essentially dimensionality reduction. Supervised, in the case of species delimitation, these are classification tools where you use labeled training data sets to inform your unknown or test data set. And you can clearly see the similarity between discovery and unsupervised and validation and supervised methods. And so this is kind of how I operate when I'm using machine learning in species delimitation projects. So I'm a postdoc looking for a job, so I have to plug my own research here. So I'm not going to talk about supervised machine learning, but I have been getting into it recently. So I have this paper we just put out. It's in review. It's on bioRxiv. But here we demonstrate an approach that combines supervised machine learning methods, which are ideally suited for species delimitation, with direct consideration of the biology and natural history of the study organisms to make more informed species delimitations. And this is particularly relevant for cryptic species, but can be applied to anything. So if this is something that sounds interesting to you, check it out. I'm happy to talk about it. But for the rest of this, I'm going to be talking about unsupervised machine learning. And this was, as mentioned previously, a paper we published in 2019, kind of showing off a few unsupervised methods and how they can be used as dimensionality reduction tools in machine learning. So here's the paper. This was a project I started as a late PhD student. It was a basic taxonomy paper, morphology, genetic data, UCE data, but it morphed into kind of more of a machine learning approach when I started my postdoc and started working with Sergey and Peter. These are, the machine learning experts. They live and breathe machine learning, and they're more on the medical side of things, but they do, they are also computational biologists. So, okay, so before I get into the methods, I'm going to give a bit of background because, despite the title of this paper, this was an integrative taxonomic revision of a particular genus of Opiliones. And that genus is Metanonychus. This is what it looks like. They are pretty small, less than 2 millimeter body size. So when you're talking about small organisms and morphological characters, this is difficult to work with. So this genus is found in the beautiful Pacific Northwest. Before this work, there was only 2 species with a number of subspecies, and in Opiliones, subspecies don't really mean anything, in my opinion. So you can find them in these moist, temperate forests where they're associated with leaf litter and woody debris. So we did some integrative taxonomy, collected Co1, morphological data, and ultra-conserved element data. What I want to show here is that it's pretty simple in terms of species delimitation. We have these kind of Co1 groupings that correspond to distinct morphological groups. These are the male genitalic morphology here. Like you can see very obvious differences, like here's the ventral plate, it's called. This one looks like a spade, this one looks like a trident, this one is a bunny ears, right? So we can see these genetic groupings correspond to morphological groups, and those are also reflected in the UCE data. So in general, species delimitation in Metanonychus was easy. So here's some basic results for some other discovery-based analyses. We have structure. It clearly shows 6 genetic groupings. 6 was favored, of course, in structure. Very, very little admixture, which is kind of typical of these organisms. And here is principal components analysis with DAPC groupings. And I'll point out here in looking at this principal components plot, right, we have 6 genetic groups, but 3 of them kind of overlap each other in this 2-dimensional principal component space. you have to look at the third and fourth dimensions to really kind of see that separation. And I'll show you that in the demonstration as well. Okay, so now onto the unsupervised machine learning. So we tested out 3 different algorithms: random forest, which you've probably heard about, almost certainly heard about; T-distributed stochastic neighbor embedding, or T-SNE for short, which you may or may not have heard about; and variational autoencoders, which you most likely have not heard about yet. So how this is going to go for this lecture and demonstration is I'm going to do a bit of lecture on random forest and T-SNE, talk about how they work and some important aspects about them, and then switch to demonstration in RStudio. After the demonstration, I'll come back, do a bit more lecture on VAEs, and then switch to another demonstration on using Google Collaboratory. So just in general, the data I'm working with here are unlinked SNPs derived from UCE data. So one thing I'll say in general for these dimensionality reduction methods is you typically want independent variables, because that's what these analyses assume. So what that means for genetic data is 1 random SNP per locus. Okay, onto the methods. So random forest, you'll hear about random forest as a supervised approach a bit later in a couple talks in a couple days. I'll give a basic general introduction to how random forest works here. So first, you need a data set. Excellent, we have our data set. This, we can just consider a SNP data set. So how random forest works is it does a bunch of what I'm going to call bootstraps for simplicity. So it takes a bootstrap of the data, and it splits that into what's called an in-bag, which is about 2/3 of the matrix size, and out-of-bag sample, which is the rest, about 1/3. So we have these 2 separate bags of data, or 2 different data sets. And so what it does is it first builds a decision tree, or a classification tree, based on the in-bag data. So this is essentially a bifurcating tree that splits all of these samples into individual units, or individual branches on this decision tree. So for example, this first bifurcation is between 1 and 2 and 3. So for example, here we would use this position A versus G, and it splits the 2. Here, splitting 2 and 3, we might use this last position, A and G. So we have our classification tree. And then what we do is we take that tree and map the out-of-bag samples onto it. And in this hypothetical example, we may get 1 up here, and 2 and 3 end up in the same box because they cannot be separated based on the data here. So for this particular bootstrap, and for every bootstrap, we calculate a proximity score. This is essentially a distance matrix, a proximity matrix. So the pairwise proximity score for 2 and 3 is 1, because they're in the same box. The pairwise score between 1 and 2 and 1 and 3 is 0, right? So it's 0 or 1. We do this over hundreds, maybe thousands of bootstraps. So we have hundreds or thousands of these proximity matrices that we then sum up and take an average. So we end up with 1 proximity matrix where each of the values ranges from 0 to 1. And this is essentially, again, a distance matrix that we can then send to multidimensional scaling, which is a multivariate technique that specifically works for distance matrices. And then we can do our clustering. So that's general and hopefully simply how random forest works. Of course, there's some differences in how you do it, whether you do it supervised or unsupervised. But this is kind of the general approach. So I'm going to switch back to the Metanonicus data set. So here again is the structure plot, here's principal components, and here's the random forest unsupervised plot in 2 dimensions. Now you can see clearly in 2 dimensions that the clusters are more clearly separated. Where principal components, you need maybe the 3rd, 4th, or 5th dimension to kind of see that. Random forest shows it pretty well in 2. There are many pros to random forest. The decision trees are uncorrelated. There's less influence from outliers. At least with unsupervised, it's pretty good with missing data. I haven't noticed anything, and I'm working with sometimes 50% taxon coverage matrices, so a lot of missing data. And it works with categorical and numerical data. One of the cons is that it can be computationally intensive when you're working with very large data sets. And the data set I'm showing here for here, there's about 300 something SNPs. So it's not a very large data set compared to some of the other more well-studied taxa. Okay, so that's random forest. Now I'm going to switch to t-SNE, t-distributed stochastic neighbor embedding. This was an approach that's very commonly used in the medical field with data sets that are thousands of samples. So I tried it with SNP data for species delimitation. So essentially what this does is it takes high dimensional data and embeds it into a lower dimensional representation, which is, well, that's what all dimensionality reduction does. But that's the general idea here. So to show you how it works, I'm going to consider two-dimensional space as high dimensions. It's so many dimensions, right? But this is how it works. You have your high dimensional plot, and what it does, it calculates similarity scores. It does this for pairwise comparisons across all samples. And it does this according to a normal distribution. So we take our high dimensional space and we calculate probabilities or similarities based on a normal curve. So samples that are close to each other will have high similarity, and samples that are farther apart will have very low similarity scores, like these ones here, right? So we do that pairwise and calculate a similarity matrix. So we can see that the squares have high similarity, the stars have high similarity, and then comparing squares to circles, a very low similarity. So this is the first step. We take our high dimensional representation and make a similarity matrix. So now we want to embed it in lower dimensions. And so going from lower dimensions, we go from 2 to 1. And so we want a one-dimensional representation of this high dimensional data. t-SNE starts randomly. So it takes the samples and just puts them into this one-dimensional space randomly. And it essentially does the same thing. It calculates similarity, but it does it based on a t-distribution. The reason it does t-distribution, or we use a t-distribution here, is because there's a higher probability for the along the tail ends of this distribution. And so what this does, it allows clusters of points to be more distinctly separated in this probability space. So this is generation 1, a random start. It calculates similarities and makes a similarity matrix. So a random start may look like this. And so what t-SNE does is it minimizes the difference between these two plots by rearranging the low dimensional representation. Eventually, after hundreds or thousands of iterations, you have a low dimensional representation that looks like this and a similarity matrix that looks like this, right? So this is how it learns. It changes, calculates, compares, changes, calculates, compares. And it keeps doing this, and that's how it learns. So what does t-SNE look like? Again, it's a two-dimensional plot. So again, principal components, random forest, t-SNE. You can see the clusters are even more well-defined. This is, of course, one of the results of using that t-distribution. It pushes these clusters farther apart. So that's t-SNE. It's pretty, for this pretty simple result, or a pretty simple data set, it looks pretty straightforward, right? We have our 6 clusters. I took this Phrynosoma data set and decided to see what t-SNE looks like with a more kind of difficult to work with taxon. So this is the t-SNE plot. From this paper, Lachey et al., I took the data set and I took the proportional probabilities. I think these were from DAPC, if I remember correctly, and I mapped them onto the t-SNE plot. You can see it makes a lot of sense how these samples are arranged in this t-SNE space, right? The more pure samples are kind of at the edges, the highly admixed samples are kind of in the middle among these different genetic groupings. So this is pretty cool. It's a good way to kind of represent, it's captured kind of that level of complexity that you don't really get from just two-dimensional space. Okay, the pros of t-SNE. It works with linear and nonlinear data that may not be so relevant with species delimitation. It preserves the local and global structure. Again, that's something that's more evident in these huge data sets, like medical data sets. But I've also done this with numbers, letters, and words. I took a morphological data set where the characters are like rounded or straight, curved, whatever. And I put it into t-SNE and it did its thing. And so here's kind of a representation of that. So here's random forest up top and t-SNE at the bottom. So I started with the structure file. I did raw nucleotides, including ambiguities, haplotypes, so phase data, and one hot, which I'll talk about when I get to VAEs, so just kind of ignore it for now. But these work with whether your data are letters or numbers, so pretty versatile in terms of the type of data you can use in these analyses. Okay, the cons, it's computationally complex. One of the guidelines for t-SNE is you generally don't want more than 50 variables, but if you have a SNP data set with hundreds, it can be kind of taxing, kind of difficult to deal with. But one way to get around that is to, weirdly enough, to do principal components analysis on your data set and just throw in only a handful of those principal components into t-SNE. But anyway, it's non-deterministic, meaning you'll get a different answer every time because it starts randomly. Of course, if your data are robust or you have even a decently robust data set, you'll get similar results or similar plots across different replicates. There are a couple of hyperparameters which are kind of subjective in terms of how you set them. One of those is perplexity. So this is that same data set with perplexity ranging from 1 to 30. And what perplexity is it influences the variance of the distribution, so that circle size. It changes the size of that circle when calculating similarities. So if you have high perplexity, that circle is really big. And your resulting plot is this gaseous cloud of samples with no structure. And as you decrease that perplexity, decreasing the size of that circle, you pick up on that structure a bit better. I tend to work in the 5 to 10 range with the data sets I've dealt with, which should be fine for most data sets. Again, unless you're doing like medical data with thousands of samples. Okay. One of the other cons of t-SNE, which may not be so relevant, again, in the data sets that we work with, is that sometimes noise will be returned as pattern. So here's kind of a random data set. When you put it through t-SNE, some of those samples are tightly packed when they're actually not. That may not be such a big problem for us, but it is something to be aware of. Now, every time I present t-SNE, somebody inevitably asks, hey, have you heard of UMAP? And yes, I have, and I've tried it. It's essentially the same thing as t-SNE. There may be some differences when you're dealing with huge data sets, but I haven't noticed any difference. It essentially operates the same way, but supposedly it captures global structure better than t-SNE. But again, not much difference. So I've showed you a bunch of two-dimensional plots for discovery-based analyses. So the next question is, how do we go from a two-dimensional plot to clustering our potential species? And that deals with clustering algorithms. I'm not going to talk too much about clustering algorithms. There are a lot of clustering algorithms. They all have pros and cons. Some work better on oddly shaped data sets, some don't, right? So it's a matter of picking what you think works best with your data. Some work with categorical, some with numerical data, right? So it just depends. The important thing is to try multiple algorithms, perhaps that vary in their kind of how they work, and then get some sort of consensus, you know. For the scripts, I picked 3 clustering algorithms. I picked them because they were simple to use. They worked in R and they didn't involve a whole lot of code. So that's what I'm going to show you. Of course, you can pick whatever you want. Okay, so I'm going to switch to the demonstration now. And for this, I'm going to share my entire screen. Give me 1 second to get set up here. Okay. Desktop one. Okay. Okay, hopefully you can see my desktop.
Shahan, can you make the text bigger, please?
Yeah, I could just, how do I do that in RStudio? Does anybody know a way to do that in RStudio?
I think in view, you can zoom in.
Okay, here we go. Whoops. Does that work?
A little more if you can.
Okay, let's do this. Okay, so this is the first demonstration. I'm going to assume there are some people who've never used RStudio, so I'm going to start from the basics. So when you open RStudio, this is what you see. It's basically a graphical user interface for the R language. I'm using R version 3.5.1. It's from 2018, which is coincidentally when I started using machine learning in R. I obviously haven't updated it. If you use newer versions, I'm sure it works fine, but you know, why update when everything works perfectly? So this is RStudio. First,
can I stop you real quick? Let's make sure everybody, I know there are some instructions to install R if you're working on a cluster, which might look a little bit different than RStudio. If you're working on your local machine, you can install RStudio. Does anybody need a link for that program, or are we all set? I don't know if is Rebecca Decal on.
Shahan, you also posted the script for this on Slack, right?
yeah, I put everything in the Slack channel. It's called PCA VAE TSNE.
And
Yeah,
it should
the
be
R script.
Yeah.
So you'll be using the R script, Metano UCE snips for rscript.str. That's the input file, and then there's the PCA DAPC RF blah 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 .r. That's the actual R script. And so you're going to want to put those, maybe make a new folder for this on your desktop, and I'll show you how to change your working directory in RStudio. Okay, should I move on? Anybody having difficulty so far? I'll try to follow along with the Zoom chat at least on the side here.
Yeah, I'll help with that. And let me say, if anybody needs help, you can post in that channel, that PCA channel, and we can try to answer questions there. You can post in the Zoom chat here. This is our first demonstration, so let's see how it goes. And again, if you're starting to feel like a little bit lost or overwhelmed, just take a step back and just listen. This will all be available. You can do it at a later time too.
So there's a question in chat about TSNE. I'll just answer it while, in case people are still. So are TSNE reduced dimensions comparable to principal components? So in terms of that space, TSNE is a bit more vague in terms of the space between clusters and things like that. You generally don't want to read too much into the distance between those clusters because they don't really mean too much. Can you tell the amount of variance? In TSNE, I believe there's a way to kind of get at that, but I haven't quite figured out in the TSNE package that I use. I think there are newer TSNE packages in R that may make it easier, but I haven't used those yet. So the question is yes, but I haven't been able to do it yet. Okay, so I'm going to go ahead and start. Okay, so setting your working directory session up here, set working directory, and then choose directory. So hopefully you have the R script and the data set in a directory somewhere. I'm operating through Google Drive for this. Okay, so this is the command to set working directory if you want to do it old school and do command line. That's one of the benefits of RStudio is that whatever you do with buttons here, it shows you what the command is. Okay, so we have our working directory. Click the Files tab. It'll show you what files you have in that directory. So I'm going to click the R script, and then it opens up a separate window with the script here. Okay, so this was a script that I've modified specifically for this workshop to make it a bit simpler. That was from the paper I mentioned previously. It's on my GitHub, the original script, if you want to check it out, but this works just the same. So if you're new to RStudio, you can install the packages with this command, install packages, and then insert package name, right? I'm just going to pick one of the newer TSNE packages. I may have installed this, but that's what you do. And it connects to the repository, it downloads it and installs it and puts it somewhere. Don't install this one. This is not the one I use. Okay. Another way to do it is this Packages tab. You can click Install. The CRAN repository is kind of the basic one, the most commonly used one. You can type in whatever you want and find it, right? So the first one we're installing is AidJeanette, or however you pronounce it. And then if you want, you just hit Install. I'm not going to do that because it may update from what I have installed already. So that's how you install. Once you install something, you have to activate it. And by doing that, you put this library and then you put the package you want. There are two ways to execute a line of code. You can hit this run button, which does one line at a time. If you're on a keyboard, I'm on a Mac, you hit the Apple key and hit enter. And then it loads the package into the memory. Or you can also check this box, whatever box you want to install. So the next one will be whatever you want to install, BibTeX, you just check this box and it installs it, or loads it. So hopefully everyone has these packages installed. I'm going to go ahead and activate everything. Okay, so all of the packages are activated now. Some of these packages may have been updated since I've made this script. I'm sure they work fine, but in case there are issues, it may be with the particular version or the R version. Sometimes there's kind of conflicts between the version of R and the version of the script, or the version of the package used. Okay, so first I'm going to show you the structure format. So which one is it? This here, this is the file. So this is standard structure format. Zoom in. Okay, so the first column is your sample name. Second column in this particular case is population assignment. And in Apiliones, every place you collect, we can consider a different population. The third column is species, or whatever kind of a priori group you want to assign. And the rest of it is actual data. And structure works with zeros, ones, twos, depending on your sort of data. And missing is -9. And this is fairly, again, a very commonly used file format. One thing to note is that every sample is 2 lines. That's because we're dealing with haplotypes here. So you can see, for example, homozygous for this haplotype, homozygous, we have this last specimen, where is it? For here, heterozygous, the 0 or a 1, right? Okay, that's structure file. Now we're going to start with PCA, principal components analysis, and DAPC. So first we have to import the structure file. Now it's this command here, read.structure, file name, here, number of individuals. It's not the number of lines in the structure file, but the number of samples. So I have 30, number of loci, 316, 1 row per individual, you're dealing with that, you would think this true, but for more often than not, it's going to be false. And then here are, I assign, right, column labels for sample label, population label. You can assign those columns here, and the rest, missing character, -9. It's going to be standard for structure.
John, what do you, are there sometimes structure data files that are formatted with one individual per row? Is that like if you just taken one haplotype out of the two for a diploid?
Sorry, say the question again.
So your structure file is formatted with 2 rows per individual, right? Do you ever see files where it's 1 row, but for example, you've just taken 1 out of the 2 haplotypes per individual?
I think I've seen that, but I've never actually used it. I don't know how that changes the results for DAPC. Maybe somebody who's done this a lot has used that one line per individual. I tend to use 2 lines, but I've seen it done. Again, I don't know how it changes the results. Maybe if you don't have a whole lot of admixture, it won't really affect anything, but okay. So going to load the structure or load the data file. When dealing with DAPC, the type of data is loaded as a gen-in format. All this stuff is here. I'm not really going to go into too much of this, right? Sample labels, locus labels, it's all kind of in there. Okay, so the first thing you want to do is, for principal components analysis, is to scale the data. This is a function called scale gen. It's a pretty generic method that scales allele frequencies. You'll see it change the heterozygous sites and for missing data. So I'll go ahead and do this. And then we have this come up here, this new data set. You can click on that and we can view the data set once it loads. So you can see, for example, that heterozygous site I pointed out earlier is now a 0.5. So this is kind of what that step does. It makes it easier for principal components analysis to work, and it also changes from 2 lines per sample to 1 line. And that's kind of what the scaling does. Okay. So we have our data set we're going to work with now. Now we're going to go to principal components analysis. So I use duty.pca. This is a very commonly used one. These are things you can adjust if you want. This is kind of the default settings I use for principal components analysis. This is the most common thing. This is something you should be aware of. This is number of dimensions you want to deal with. Okay, so this does the principal components analysis. I'm going to do that, and it gets into this, put it all into this variable here, PCA1, and we can take a closer look at that. Here's the results of principal components analysis. We're going to be dealing with this one here, which has the principal components. You can either, here, I think there's a scaled one if you want to deal with that. This is the scaled one, but principal components are all stored here. So now we want to view the principal components. Yeah, so I'm just going to plot this really quick. Here's the principal components. This will come up in your plots thing. You can see it here. I have a function here for if you want to label the samples based on sample name. I like to keep the plot and the samples different. So I'm going to get into DAPC. I'm going to go through this a little bit quicker, I think. So first, clusters is the first step of DAPC. What you want, this determines the optimal number of clusters. So you have to include a certain number of principal components for this step. It's fine to include all of them. So I'm picking 30. And then here's our BIC plot with the optimal number of clusters. All right, the lowest one is optimal, which is 6. So I'm going to choose 6. This variable is stored.
Can I ask a quick question?
Yeah.
So when you're choosing your max M clusters, how do you decide on that? Just because I've used different numbers there and gotten really different results for DAPC.
In this particular data set, I do not see much different. However, if I picked 29, it would heavily favor 29 because of the problems with working on organisms like this. So I just arbitrarily set it at 20 so I don't have to deal with that oversplitting, right? So that's something that happens a lot even in basic things like this. So you can change it ideally to be purely discovery. You want to set it to the maximum number of samples or populations you have. But I did not do that because I don't want to deal with that 29, right? There are 30 samples, but they're from 29 different populations, this data set. Okay, so group K, this just stores that number in this variable here, which I'm going to call later. Next, we're going to do this assigns those, so this determines the number of clusters, and then the rest of this assigns the samples to those clusters. This step, you don't want to pick all of them, you want a little less, you want to do, or you don't want all of your principal components. So I'm going to pick 5 because I know ahead of time that that's the best number. And then here, again, there's objective ways of picking this. I'm just going to pick, I don't know, 3. Let's just go with 3. And then here we're going to view our results. Here's one plot where you have your samples here, and then it gives you assignment to whatever cluster, right? It's not pretty because I haven't organized by voucher number, not by species. Here's another way to view it. This is proportional probability. If you were dealing with a more complicated data set, you would see proportions, but because I'm dealing with the Pileones, it's all 100%. Okay, this just plots the principal components with those DAPC clusters. So you can see there's a lot of overlap here in 2-dimensional space. I'm going to quickly, maybe don't try this if you're on a PC, but this is for, I'm just going to quickly show what it looks like, what the principal components plot looks like in 3-dimensional space. So this is what it looks like. We'll be looking at it, like this. So this is what we're seeing here. But you can see if you look at the 3rd dimension, some of these other ones become a bit more separated, right? Okay, so that's principal components, DAPC. I'm going to switch to random forest, unsupervised random forest. So we have to convert the data. I'm going to start with the scale data, and we have to convert it to factors to work with random forest. So I'm going to do that really quick. These commands may or may not actually do something to your data set. These are just the cumulative commands that I've used based on the very different data types. Again, attempting morphology, whatever. I just do all of them no matter the data type, and it converts it just fine. This is number of samples. I just put that as a variable. So this is random forest. As complicated as random forest is, this is all it takes to do unsupervised. I'm going to start it while I talk about these variables. Okay, RGL works on PC. Yeah, I haven't tried it on PC. I just like, I know you have to install the Xcorts, whatever. Okay, so there are 2 variables that you need to be aware of. N tree, which is the number of replicates you do. I set it at 5,000. It's maybe a bit too much. And then M try, which I leave at default, but that's this M try. That's the number of variables sampled in the tree. I leave it at default, that's fine. So we have a random forest, a bunch of stuff here, importance, which is something important. And then this is the proximity matrix here. So I'm going to send this to multidimensional scaling. This is, again, specifically for distance matrices, distance data. I call this function MDS plot is a function within the random forest package. So it deals with proximity instead of basic distance. I'm just going to, okay, this may not work with the scaled. I'll try it again. If you get this error, yeah, it's not going to come up. I'm sorry, I have to reduce the size for a second here. Oops. Okay, the reason it throws that error again. Okay, the reason it throws that error is because this, right? This is showing all the principal components You don't want all of them. I just want to retain a few of them. This is what's called the broken stick method. This is something I took from the Ezard et al. Paper. It's just a way to objectively determine the number of principal components to retain. And in this case, it picked 5, as I mentioned earlier. So let's zoom in again. Okay, so now we get into clustering. Although first, I'm going to plot random forest. So here's the random forest 2-dimensional plot. It's not exactly like I showed in the lecture, but it's fine. So random forest with DAPC clusters. If you want your labels, it's there. I'm going to go through this part really quick. These are 3 different clustering algorithms: PAM clustering, gap statistic, and hierarchical clustering. They should ideally all give you the same. DAPC, you have to specify your K value ahead of time, in this case 6. And what PAM does is you specify the K value and it assigns those samples. And of course, it's exactly the same as what I showed previously as DAPC. If you want to do kind of discovery-based PAM clustering, you can use this function and it calculates the optimal PAM clustering based on a specified range. In this case, again, it's 6, no surprise there. Again, we can plot those. Most of these are just plotting functions. Gap statistic is another one that's a little bit different. It uses k-means clustering. You can, yeah, there are some variables you can set, but for the most part, it's pretty standard. Does a bunch of bootstrapping. This is how it uses, what it uses to determine the number of clusters, right? The gap between within cluster distances. So it picks 6. That's surprising. Last time I did this, it picked 7. So we're not going to get any variation here. And then plot it, right? It's exactly the same as DAPC. Hierarchical clustering, another form of clustering. You specify your K values here. I just do 10 to keep it simple. This one picks 7, so we get our variation. And when we plot those clusters, it splits one of them in 2. That's fine. Okay, on to t-SNE. So I'm moving quick. I want to make sure I get to VAEs, which are objectively way more cooler. So t-SNEs, you have to do a bit of functions for kind of prepping the plot labels and colors and such. These are kind of standard for R, picking colors and names. This ECB is specific to t-SNE. It's a callback function. You'll see when I run t-SNE, you'll actually see it happening. So this kind of helps with that. If you have population assignment in your structure file, you can use this instead, and it'll color that based on your population assignment. This colors it based on DAPC clusters. Okay, here's t-SNE. I'm going to do it on the principal components analysis because it goes a little faster. You can also do it on your data file if you want. Max iterations, this is number of generations. Here's your perplexity. This is where you change that perplexity, dealing with 5. The other important one is K. This is the number of dimensions you want the embedding. I want 2 dimensions. So t-SNE runs and you'll see it learning, right? The samples are moving around. It's calculating the similarity and comparing, and it's moving samples around into this lower dimensional space. After what, 2,000 generations, it doesn't really change that much. For those of you who can't see yellow very well, there is a cluster here. But we see our 6 clusters in this space very easily. Okay, 5,000, it's done. I'm not going to go through these clustering algorithms for the sake of time. It's essentially the same thing, but instead of using, the RF output, we're calling the t-SNE output, which is the first 2 dimensions, or essentially the XY placement of these samples. Okay, and the rest of this is output files to make it nice and easy. I'll just do this first few just to show you. And everything you need when you're kind of making your publications. Where'd they go? Somewhere. Okay, like here's the principal components one. Basic plot, clusters, samples, right? And then your loadings are here, or wherever you have your output. So that's basically it. You know, for as complex as random forest is and t-SNE is, it was kind of surprising to me how simple it is to execute, right? Random forest is just this. Okay, so I'm going to switch back to lecture. If there are questions, I'll try to answer any questions. Is there a good resource for different clustering methods? I'll show that. I'll go back to lecture and answer that. Okay, let me stop share.
Hey, we can also, there is a break after, a lunch break for bioinformatics support, but you can create a breakout room if you can stay and answer some questions too. That would be great if we run out of time.
Okay, so we're back to PowerPoint.
Yeah, looks good.
Okay, so to answer the clustering questions, I'll talk about this website more, but honestly, go into Google and type in clustering algorithms and you'll find something. I like Towards Data Science. They have some good visualizations. There's this paper, which I, looked at pretty quickly, but it has a nice table of pros and cons and how to use them and things like that. So there are resources out there. Here are the two that I found explains it pretty well. Okay, so switching to VAE. Variational autoencoders are a type of neural network, and they fall into the range of machine learning that is deep learning. And what deep learning is a way to learn successive layers of increasingly meaningful representation. So the deep in deep learning is exactly that. You have your original input, you have your layers, you have your final output. The best explanation of this I've seen is it was called a multi-stage information distillation function, where your information goes in and through these successive filters, it comes out increasingly purified. I don't like that word, but it works here. Okay, so that's the deep in deep learning. The learning part is represented like this. So this is here. We have our input, layers, and our output. These layers are controlled by weights. They are specifications of what a layer does to the data. It's basically a transformation that's parameterized by the weights. We get our output and we compare it to our true targets. And we calculate we calculate a loss function. This is exactly like it sounds. It's how far the output is from expected. We get our loss score, which is part of what's called back propagation. So it uses this loss score, some optimizer uses the loss score to adjust the weights and how they transform the data to minimize the loss in the next function. So that's the learning part. There are many different types of deep learning. You've heard convolutional neural networks. The VAE is a type of autoencoder. Very simply, original input, it goes through some layers that encode the data to a compressed representation. And then it decodes the data, essentially does this backwards and attempts to reconstruct the input based on this compressed representation. And then of course it compares. Variational autoencoders are a type of autoencoder, but generally how it works is like this. We have our input data, whatever these are, right? We put it into the autoencoder, and in this case, our latent space or reduced dimension space, it learns 2 dimensions, cat face. And then what it attempts to do is it reconstructs the original data based on this. So maybe after a few generations, it looks like this. It's not good. I drew this while I was eating a sandwich and with my left hand. So it's not nice, but it's a good start. After maybe 100 generations, it looks like this. It's not ideal, but it's certainly better than the sandwich cat, right? After hundreds or thousands of generations, you get this. This is certainly one of those. So that's essentially how it works, by comparing the output to the input. What makes the variational very special is what it does in that latent space. It assumes the data are generated from a statistical process. And it produces 2 sets of values, the mean and standard deviation of those samples. So what that means is that each sample in this latent space has a mean of 0 and standard deviation of 1. All this is derived from Bayesian probability theory. So this is like ideally suited for, for systematics, right? Okay, so what does the output look like? Here is the mean. Again, two-dimensional representation, everything is clearly separated, but the benefit of VAE is the standard deviation. Here we can see the clusters. I interpret this as the uncertainty around those samples in this latent space. Now with this data set, it's very simple, not complicated. But when we look at a more complicated data set, for example, these fringed toad lizards, where there's a bit more going on, we see where the standard deviation comes into play. So we look at these three species here. Rufo punctata was, we heard about this earlier, a hybrid, identified as a hybrid population. So you can see it falls directly in between the two parental species and the standard deviation overlaps compared to the other approaches where principal components analysis didn't even identify this hybrid. RF around the forest did a little better, but not perfect. And TSNE had very similar representation, but again, standard deviation. This approach has been used quite a bit in various taxa that vary in their number of legs. People are finding it useful. So really quickly, this is our model. I'm not going to talk too much about this, but there are various things that go into this, like dense just means that it's a fully connected layer. Every neuron in the second layer is connected to every single neuron in the first layer. Dropout is something I'll mention. This is randomly drops out or randomly deactivates 50%, in this case, 50% of the neurons. It's a way to prevent overfitting activation. This is essentially a way to send the information from one layer to the next. Batch normalization is just normalizing the data. Two things to be aware of with the VAE, you have your loss function and your accuracy. So as it's running, the loss decreases, of course, because it's learning and accuracy increases. One thing that's very cool about the VAE is this is a VAE as it's running the data set. One thing I noticed, it happens very quickly at the beginning, but the way it separates those samples reflects the phylogeny, right? The deepest split happens first. So that was something very cool that we found by using this. The pros, all the benefits of neural networks, you know, complicated data, you can model very simply. The best benefit of VAE specifically is the mean and standard deviation. However, this is a black box. We don't actually know what's happening in here. We don't know what it's finding important. There are people doing this, of course, to kind of get at what it's finding important. There are many parameters you'll see, and it's non-deterministic, right? It starts randomly, and across replicates of the same data set, they never look exactly the same, right? So here's 3 different data sets. They never look exactly the same, but the way I pick one is just like this. We have our loss function. I treat this loss curve very much like the likelihood curve in a Bayesian analysis. I calculate a burn-in, in this case 50%. I cut that out and I calculate the average loss for that second half and I compare it across replicates. And the one with the lowest loss is the favored representation. One thing about VAE is the input format. I do what's called one-hot encoding. This is a very commonly used input format in deep learning. Essentially what it does, it changes categorical data to integer data. So in this example here, we have 3 options. And so for every single, so red gets translated to 1, 0, 0, green is 0, 1, 0, right? This is for categorical data when no ordinal relationships exist, and it changes it to essentially binary variables. So for nucleotide data, this is what I do. Because there are 4 nucleotides, I have 4 binary variables. This is exactly what I do. If you're starting with structure, this is also what I do here. Missing or N is all zeros. This, of course, can accommodate ambiguities. For example, if you have an A or a C, becomes 2.5s. Okay. The other benefit about one-hot is that the distance between any 2 variables is always 1. Okay. Switching again to demonstration. So this is going to be in Google Collaboratory. So if you haven't used Google Collaboratory, the benefit of this is that you don't need to install anything. Where did, okay. Stop one. Okay, so hopefully you can see. Is that the text size okay for this?
That looks good, yeah.
Okay, so you type in Google Colab, click this button, and you'll get this window. It knows where my files are, but for the file I gave you, you're going to go to this upload, and then you can just drag and drop, where to go? This file, VAE, workshop, drag it in there.
Just let me jump in real quick. That file again is pinned on that Slack channel for quick access.
I didn't hear anybody using Kaggle, so I'm just going to not do that. If somebody does not want Google involved in any of your work, you can use Kaggle with this this script. There's a couple minor modifications. I can send you this script if you want to do that. Okay, so again, this just uses the Google backend. You don't have to install anything. It's amazing how this works. So I'm going to go ahead and open this here. Okay, is there a script to convert a file into VAE? So yeah, I have some here. Depending on how your input data works, I can do this. I just use, I'll show you really quick. There are functions built into some of these packages. TensorFlow and Keras are machine learning packages that will convert your data, but I just use very, very not pretty script, but set, basically search and replace. I can send you examples if you really want to do that. I've also done it by hand in like BB edit, you know. Okay, so how this works is this is what's called a Jupyter notebook. You break your code down into different boxes and you can execute one at a time. You just click this arrow button and it executes. This first step is loading everything into all the packages into memory. So it uses, again, TensorFlow, Keras. These are machine learning packages. You might be, you should be familiar with NumPy and Matplot, Seaborn. I use colorblind so that people can see the colors better. Okay, so let me show you one hot format really quick. So this is one hot. I always do that. Okay, hopefully that's enough. First column is sample name. Second column is species or a priori grouping. Columns are separated by spaces. Here is the first nucleotide of the first sample, right? For this case, I use 0.0s, not just 0 and 1, right? Because we're going to deal with, when we have heterozygous samples, we have our 0.5s, right? So this is one hot format here. This is what it looks like, very simple. So again, this is something you can easily do, just search and replace. Okay, so packages are loaded. This just functions for saving and loading the data, right? The name, group, one hot. This is column 1, column 2, the rest of it. Some basic functions for plot generation. I'm going to go through this quick. I apologize. People have specific questions. We can talk about it in the hour after this. This plot is for the Z space, the latent space, essentially the mean of the samples. This is for mu and sigma, or the mean and standard deviation. And we chose 100 plots or 100 samples of that distribution space for each sample. Here's the VAE model. There's a lot of variables here. Dimensions, number of SNPs, it'll take that automatically. Number of categories will be 4. Latent dimensions should be 2, right? That's number of dimensions you want in your latent space. Here is the encoder and decoder. What you see here, 3 layers, each of 100 neurons. For the encoder and decoder, dropout is 0.5 for each layer. These are the activation functions. And then this is the encoder, right? Handles the input. This is the encoder with our dense, dropout, activation, batch normalization. Here's our latent space with mean and standard deviation. And then we work backwards, decoder and output. And then here is where we calculate our loss and accuracy. I don't have time to go into this, but there's two measures of loss, KL loss, and then the reconstruction loss, both of these. This compares, yeah, they both go in together to return the overall loss function. And then accuracy, it's all here. Okay, so here's more. It's really complicated. Functions to generate the models, train and plot the latent space. The important things here are right here. So batch sizes is something you can adjust as well. We test multiple batch sizes. This just is how much of the data to subsample when it's working through the VAE. Epochs. This is generations, equivalent to generations. So I have 1,000. It seems to work fine. One thing I'll note is that all these parameters, we didn't just pick the best one for this data set. We had 5 or 6 different data sets in addition to a set of simulated data sets where we found the best set of variables across those. And I showed you some Pilioni stuff, there was some lizard stuff, and of course, simulated data. So these should work for the majority of data sets you attempt this with. Okay, then we're going to import the data set. This function calls it directly from the GitHub page where we have our VAE stuff stored. This is Sergey, my collaborator. This is his GitHub. So we're going to call that, and it should return ls. This is basic Unix. Return the data set. If you click this refresh button here, it'll show you that data set in your working directory. Okay, this is where it runs. Click go, and you will get some warnings. Every time I do a set of analyses, I get warnings. It's still running, right? You see this thing spinning. It's telling you how long it's taking, what it's doing. This just means some of the functions are deprecated and they'll be replaced. I don't worry about it until I get a fatal error. It's working, it's doing its thing. So it takes about, again, for a data set of 300 SNPs, it'll take about a little less than 2 minutes. So it's not too long. So while we're waiting, let's see if there's questions. Yeah, the first A is 1000. Yeah, it takes that one hot. It doesn't, when you put it into the VAE, it doesn't read it exactly like that. There's some functions to condense that into kind of two dimensions essentially. So it converts that into an easier interpretability for the VAE.
I think there's a good question from earlier. What do you do if the clustering algorithms do not agree on the number of clusters? This was from the beginning of your talk.
Well, if they don't agree, that's fine. You got some discordance and you have to explain that biologically or algorithmically. So it's also in terms of discovery, if you get different number of clusters, those are then different competing hypotheses that you test in validation. So there's nothing wrong with getting competing or different answers when you're doing clustering. And of course, if there's some obvious assumptions about the clustering algorithm that don't fit your data, you should throw it out or not even use it. But I consider it as more hypotheses to test. Or maybe there's something in your data set that you didn't see first time. Okay, so here's our basic output. Loss curve, accuracy curve. Here, oh, one thing I didn't mention, I'll go back. This is the loss of that particular run. So I mentioned the burn-in and whatnot. That happens here. We have our loss history, accuracy history. I put in this. This is not in the original script. This is something I put in afterwards. This is where you calculate the average loss after 50% burn-in, and that gets reported right here. So after you do this 5, 10 times, which is what I do with any data set, I pick the one that has the lowest loss. that becomes the main figure and the rest go into supplemental. Here's the basic output, mean, standard deviation. This is all the output into files. A CSV file with the XY scatter for each sample, the mean and standard deviation, and then data labels, sample labels. You click refresh, you'll see all that here. And to download, you just click this button, download, or for PDFs, you just double click and it will download to wherever your internet files get downloaded to. So here's what's output, the mean, standard deviation, and samples. So that's VAE. Again, discovery approach. You can then take this, your plot here, and do whatever clustering you want, downstream clustering. But again, this is pretty good visualization of what's happening in your data set, very simply. So I have a few minutes left. I'm going to switch right back to a couple last slides. No, yeah, sure. Okay, I think we're back to. Okay, so helpful resources. Very recently, this cool paper came out. It's a review paper, Deep Learning as a Tool for Ecology and Evolution. It's a preprint, a very good start. If you want to learn about VAE and convolutional neural networks, which Brian mentioned as well, this is where I would start if you know nothing. If you want to get a bit more specific, I use Deep Learning with Python. This is a cool book. There's also a lot of free online code he put up if you want to start getting into the actual code of it. The more generally with machine learning, I mentioned towards data science. This was, you know, when I go into Google and I type in what is an activation function, more often than not, towards datascience.com is where I find a good answer, right? Everybody learns differently, everybody teaches differently. Towards data science has like multiple people can contribute. So more often than not, this is where I go. I'm currently working with Devin DeRad to develop an R package that does all of this. He works on birds. So I don't know much about birds other than they're nice to look at. But he, in his recent publications, has been turning this into a script, and we are going to develop an R package that goes from data to very nice figures like this. So I think I'm right on time. So thank you for your time. Thank you for the organizers, Andrea, for inviting me to do this. I'm happy to help and answer questions. I'll work through the questions through the break, or if you want to try these out in the future, my email and Twitter account are here. Please feel free to contact me again. I'm happy to help and send you kind of data formatting scripts if you want. So again, thank you for your time, and I'll answer questions.
Thank you so much for that. It was a really nice overview. I have a quick question about your Jupyter Notebook file. Is that easily convertible? Like I could use that and input my own data pretty easily? Or is that what you're hoping to do with this new R package that you're developing?
So that's something we're going to have to talk about, converting this Jupyter script to R. There's actually very recently VAEs were implemented in some R packages. I haven't looked at them yet to see how versatile they are, but I think that's something we're going to have to figure out for the package, the R package. We're either going to have to do it outside of R and then bring in the data set, or make a new VAE completely in R. Oh, wow. So that's something that's still we're working on, but I will mimic it as close to what I've done with what we've done with in Python.
So there's a couple more questions in the chat. I think Inbar had a question about how you make decisions about setting some of the components, the parameters, et cetera. And maybe we can follow that up in the Slack, so everybody will have access to that. Somebody asked, will phase SNPs work with these approaches?
Oh yeah, yeah. One of the when I was showing the different formats like letters, numbers, one of those was haplotypes, phase SNPs. And when you do it that way, you get twice as many points, right? You have 2 points per sample, but it works just fine. There was, compared to kind of doing it through DAPC, it was a bit blurrier in terms of the species boundaries, but that's kind of, you're capturing that variation a bit better, I think, when you're working with phased SNPs. But yeah, it works the same. Yeah, and I can answer the rest. Yeah, the question about decisions for setting parameters, we can handle that in Slack.
That's great. Any other more general questions?
Why don't I switch to studying fishes? I'm to the core of my soul, I am an arachnologist. I study arachnids. I'm not opposed to helping out other people in other organisms, of course, but the reason I work on arachnids, and specifically Apilionids, is because there's still so much left to do. Again, I have these, there's so many undescribed species. Fish are cool though. And if there are any botanists watching that are doing species delimitation, I'd be happy to collaborate, because there are some cool things we can do with machine learning and plants. I think combining genetic data and kind of bioclimatic data, because that's something I'm also interested in as well. So if anybody wants to collaborate, whether you work on arachnids, fish, or plants, or anything, just I'm happy to talk.
I'm pretty sure we have a few botanists in the chat room, some fellows
Nice.
and research staff.
Nice.
All right, well, great. Well, we have an hour break. I want to thank all the speakers this morning. It's been a great morning or evening or afternoon, wherever you may be. I think for the next hour, let's see if Vanessa is online. Yeah, Vanessa Gonzalez will be here to help with bioinformatics related questions. Maybe Sean can stay on as well. We can talk about this, use this time to think about the next session, which isn't really a lot of installation, which is good for some people. And maybe we can also get this to work on some of your systems as well, what we had just discussed. So thank you all again. And I will be back in 10 minutes or so. You can use this time to join this discussion or this breakout room or take a break. The next speaker will be in an hour. Thanks.
If you have any installation questions, you could add them to the troubleshooting channel, the general channel, or you can add them in the chat if you guys need any help.
Vanessa, is there anybody available for like one-on-one troubleshooting for specific issues?
Yeah, if you send me a message, that's what I have been doing. It's yeah, so if you send me a message, I can try to troubleshoot one-on-one. If not, I could point you to other folks that might be able to help as well.
Great. Here on Slack.
On Slack, please.